Hello everyone, this is the same material that you have and I'm providing some comments on them. So the first thing we need to do is go through the objectives that I expect you to get from reading the material. I should point out that the materials you have, the handouts, should be enough, but the comments I'm providing is just for explaining more concepts and more comments on the figures. Um, so you should be only responsible for the handouts and the material. Any comments I add in this video is basically optional or just for further explanation. Okay, so let's start with the reading objectives. The first one is I expect you to become familiar with the types of drug targets and examples of each. I expect you to understand the chemical nature of proteins, in particular, as an example of drug target, because they are the most common type of drugs. There is a video that I implemented which gives you a brief introduction to protein structure. I expect you to understand the overall process of the receptor-ligand interaction, including the formation of the intermolecular bonds, the conformational change to the active site, which is known as induced fit, followed by the signal transduction, which produces the biological effect, and finally departure of the ligand from the binding site. You should be able to differentiate between an endogenous ligand binding site and an allosteric binding site. Also differentiate between the different types of intermolecular bonds and be able to rank them by strength, particularly the first four types, which are the covalent, ionic, hydrogen bond, and van der Waals interactions. I expect you to relate the impact of the strength of the intermolecular bonds on affinity and how to define the affinity and how is it measured, and relate the impact of intermolecular bonds on the induced fit and how this affects the biological activity, how some agents can produce the same induced fit and thus produce the same effect, while others do not produce the same induced fit and thus block the receptor. Also relate the structure to the activity and differentiate between 3D pharmacophore and the generalized Born pharmacophore, and understand the similarity principle and how it could be used to identify the pharmacophoric groups. Finally, recognize the structural features such as optical isomers, geometric and conformational isomers, differentiate between these three kinds, and understand how these features can impact the pharmacodynamic properties and thus the pharmacological activity through the example I provided for each kind. And then, as you expect, there should be an IRAT and a TRAT on this material, and we'll have some applications. These references are just optional readings. In order to design painkillers, we need to understand how the pain receptors are activated and how they interact with natural chemical messengers, also known as endogenous ligands. The idea of having a receptor that binds to the natural chemical messenger or to the drug was first developed by Ehrlich, who explained the biological action of molecular entities. He postulated that receptors are certain side chains that are located on the surface of the cells, and these side chains are complementary to the chemical messengers or the endogenous ligands. The two, which is the side chains and the chemical messengers, could therefore, since they are complementary, could therefore interact with one another and produce the biological action. Um, so let's understand these. In order to understand these side chains, let's explain the chemical nature of these receptors, or also known as drug targets. Okay, so drug targets can be classified based on their chemical nature into four groups. There could be lipids, such as cell membrane lipids. There could be nucleic acids, such as DNA or RNA. There could be carbohydrates, like the cell surface carbohydrates and antigens. And last but not least, there could be proteins, which is the most common type of these drug targets. And these proteins could be structural proteins. These are proteins that are involved in building the structure of the biological system, such as tubulin. They could be transport proteins that are responsible for transporting polar molecules or amino acids. They could be also transmembrane, and there are three kinds of transmembrane proteins. There could be an ion channel protein, transmembrane, which means that they are attached to the membrane of the cell, and they have some part of them as outside of the cell and another part inside the cell, so they are known as transmembrane proteins. So they could examples are again ion channels, or the G protein coupled receptor, a very important protein, or receptors that has enzymatic cytosolic domains that could catalyze reactions such as kinases. And they could also be cell surface adhesion receptors. They could be intracellular receptors, and they could be extracellular enzymes, such as the enzymes that catalyze the reactions inside the cells, such as esterases, for example, which are an example of extracellular enzymes. Since proteins are the most common type of drug targets, we will review the chemical structure of proteins. I know some of you have not taken biochemistry course, and to make sure everybody is on the same page, I provided a video for you to watch. So just click on this video, and while you watch the video, make sure you are able to answer the following questions, which is what roles do proteins play in biological system? What are the building blocks of proteins? What type of atoms are involved in these building blocks? And what is the difference between primary, secondary, tertiary, and quaternary structure of a protein? So let's go ahead and watch the video.
Proteins play countless roles throughout the biological world, from catalyzing chemical reactions to building the structures of all living things. Despite this wide range of functions, all proteins are made out of the same 20 building blocks, called amino acids. The way these 20 amino acids are arranged dictates the folding of the protein into its unique final shape and its function. Amino acids are made of carbon, oxygen, nitrogen, hydrogen, and sulfur atoms. These atoms form an amino group, a carboxyl group, and a side chain attached to a central carbon atom. The side chain is the only part that varies from amino acid to amino acid and determines its property. Hydrophobic amino acids, such as leucine and isoleucine, have carbon-rich side chains, which don't interact well with water. Hydrophilic amino acids, such as serine or threonine, interact well with water. Charged amino acids like glutamic acid or arginine interact with oppositely charged amino acids or with water. The primary structure of the protein is the linear sequence of amino acids as encoded by DNA. The amino acids are joined by peptide bonds, which link an amino group and a carboxyl group. A water molecule is released each time a bond is formed. Specific amino acid sequences give proteins their distinct shapes and chemical characteristics. These protein chains often fold into two types of secondary structures stabilized by hydrogen bonds. A protein chain can fold into a rigid alpha helix, forming regular patterns of hydrogen bonds between the backbone atoms of nearby amino acids. Backbone atoms of the chain can interact side by side to form beta sheets. Many proteins fold into a compact globular shape, with hydrophobic side chains sheltered inside away from the surrounding water. The functions of many proteins rely on this folded structure. For instance, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds oxygen. Two or more polypeptide chains can come together to form one functional molecule with several subunits. The four subunits of hemoglobin cooperate so that the complex can pick up more oxygen in the lungs and release it in the body. Many proteins rely on the ability to recognize the shape of specific molecules in order to function correctly. The flexible arms of antibodies protect the body from disease by recognizing and binding to foreign molecules, and thus preventing the viral RNA or DNA to enter the cell. Collagen forms a strong triple helix that is used throughout the body for structural support. The calcium pump moves ions across cell membranes, allowing the synchronized contraction of muscle cells. The hormone insulin is a small, stable protein that can easily maintain its shape while traveling through the blood to regulate blood sugar level. Alpha amylase is an enzyme with a catalytic site that begins the breakdown of carbohydrates in our saliva. Ferritin forms a hollow shell that stores iron from our food. Learn more about the functions and 3D structures of nucleic acids, proteins, and molecular machines at the RCSB Protein Database.
Okay, so now since we have a better idea about the structure of the protein, in this figure we see an example of how a protein here is interacting with the drug. Here is a protein, and these are the side chains, amino acids of the protein in blue, and this is a drug in orange, and these groups from the drug and from the amino acids of the protein are complementary to each other. They are interacting with each other. The drug should fit this binding site. It should have the proper shape, and it should also have the proper intermolecular bonds, which we will discuss soon, that represent the way these function groups are interacting with each other. Okay, so let's look at the overall process of the receptor-ligand interaction. If your receptor is a protein, it would look like that, which is a chain of amino acids. This would be the binding site, which is a hydrophobic hollow that is nearly the correct shape for the natural chemical messenger. At this surface, you will see the amino acids, which is the side chains for a protein that should be recognized by the chemical messenger. Sometimes a receptor might have another binding site that is different from the binding site for the natural chemical messenger. This binding site is not recognized by the natural chemical messenger, but it's another site that a ligand or a drug might bind to and affect the activity either positively or negatively. This binding site is referred to as allosteric binding site. The binding process starts by the binding site accepting the messenger and binding to it through the intermolecular bonds. By these intermolecular bonds, the shape of the binding site will change such that the bonds become stronger. The change in this shape in the binding site is referred to as induced fit. Notice how the bonds here got stronger and the shape of the receptor has changed from this to that here. And notice also how as a protein we have some amino acids such as serine, aspartate, and phenylalanine binding to the messenger here and making these bonds stronger. The resulting induced fit maximizes these intermolecular bonds and results into something called as the signal transduction, which is a signal that's transferred inside the cell to produce the biological effect. The signal transduction is similar to an effect called the domino effect, where you activate one protein, for example, and then this protein leads to activation of the other proteins, leading into the series of signals transductions, which leads to the biological effects. Notice that the messengers do not enter into the cell. They only bind to the surface, and then they leave the receptor unchanged and don't stay bound permanently. We should also notice that the binding sites are so specific such that any small change in the messenger can affect the induced fit, and this can affect the signals transduction and can affect therefore the biological effects. The binding interactions should be strong enough to hold the messenger into its receptor such that it produces the signal transduction and the biological effect, but it should also be weak enough to allow the messenger to depart the receptor. Therefore, there should be a fine balance between for the interactions of the messenger with the receptor. It should be strong enough, but at the same time weak enough. If we design a molecule that binds to the receptor very strong but does not leave the receptor, then this molecule is going to block the binding site, and we will have a molecule that we refer to as antagonists, which we are going to discuss later. Now let's talk about the types of the intermolecular bonds that can occur between a ligand and its receptor. The first one is the covalent bond, which is actually not commonly used because if it binds into the receptor, it would be very hard for the ligand to leave the receptor and will produce an antagonist as we described earlier. But it is the stronger kind of bond. The energy that it takes to break this bond ranges from 150 to 1000 kilojoule per mole depending on the bond type. Triple bond would be stronger than double and stronger than single, and it also depends on the atoms involved in the bond. Covalent bonds are sometimes irreversible, and they are used to produce ligands that are designed to bind and not leave the receptor, such as toxins, poisons, chemotherapeutic agents like antibiotics. In this figure here, we have an example of how a covalent bond is formed. Just for example, if we have an OH group here in the binding site and we have a leaving group here in the messenger, so the binding occurs and the covalent bond here is formed. Again, this is not a commonly used bond unless you intend to have the messenger to permanently bind to the receptor. The second type of intermolecular bonds is the ionic, also known as electrostatic bond. It is the second most stronger intermolecular bond after the covalent bond we described earlier. The energy it takes to break this bond ranges from 20 to 40 kilojoule per mole, and the ionic bonds occur between chemical groups of opposite charges. For example, if you have an amino acid here with a positive charge on the nitrogen and you have another carboxylic acid here on the drug with a negative charge, then we will know that the bond here is an ionic or electrostatic bond. The opposite can occur here. You might have the positive charge on the drug and the negative charge on the target, for example, on an amino acid. And again, you will have an ionic bond, another example of an ionic bond. As you can imagine, the closer the ions to each other, the stronger is the bond, and as the distance increases, the bond becomes weaker. Ionic bonds are considered the most important initial interactions as the drug enters the binding site because they have a long range of interaction. The third type of intermolecular bonds is the hydrogen bonds, which are weaker than ionic interactions, but they are stronger than van der Waals interactions, which we will see after this one. Hydrogen bonds occur between electron-rich heteroatoms such as nitrogen, oxygen, or fluorine. Nitrogen, oxygen, or fluorine, an electron-rich heteroatom, and an electron-deficient hydrogen that is attached to another heteroatom such as oxygen, nitrogen, or fluorine. The heteroatom that's attached to the hydrogen is known as the hydrogen bond donor, and the other heteroatom is known as the hydrogen bond acceptor. What's unique about this hydrogen bond is that it's directional, which means that the angle between the heteroatom, the hydrogen, and the other heteroatom should be close to 180 degrees in order for the hydrogen bond to occur. The fourth type of intermolecular bonds is the van der Waals interactions, which is the weakest among the bonds we discussed so far. It has a bond energy of 2 to 4 kilojoule per mole. Van der Waals interactions occur between hydrophobic regions of the drug and hydrophobic regions of the binding site of the receptor. The way these bonds are formed is when a transit change or when a distribution of the electron density occurs in one hydrophobic region and another disturbance and the electron density occurs in the other hydrophobic region in the binding site. These partial positive, partial negatives charges will result and attraction occurs between the hydrophobic regions. As you can imagine, these van der Waals interactions require that the hydrophobic regions be very close to each other, so they depend on the distance. If the distance is large, the van der Waals interactions will not occur. Although the van der Waals interactions are very weak, but they are very crucial to the binding process because of the cumulative effect of the overall van der Waals interactions from the drug and the binding site, from different parts of the drug and the binding site. Please differentiate between van der Waals interactions and what's called hydrophobic effect. Hydrophobic effect is what happens when nonpolar molecules aggregate in an aqueous solution in order to separate from water and minimize its exposure to the water polar molecules. Hydrophobic effect normally occurs between hydrophobic regions of the drug before van der Waals interactions take place, meaning that before the drug approaches the binding site, they have to get rid of the water molecules and they aggregate with each other. These hydrophobic regions aggregate to each other before van der Waals interactions occur. Okay, so after we have studied the most commonly used kind of interactions, there are other kinds of interactions that I'm going to list here. The first one is called ion-dipole interactions, occurs between an ion and a polar molecule. Here's the dipole. We have a partial negative, partial positive being attracted to an ion. Another example here, if you have a positive ion for an ammonium, it's attracted to this dipole, so it's called ion-dipole interactions. Now, if you have a polar molecule here as part of the receptor site, and you have another polar molecule here as the drug, then a form of bond called dipole-dipole interaction can occur, as in this example here. So the molecule will align such that the positive charge, the partial negative with the partial positive, and the partial negative with the partial positive. The third example here is for the induced dipole interactions. If you have a dipole or a polar molecule here as a drug, and you have a hydrophobic region like the benzene ring here as part of the binding site, then this dipole molecule can induce the hydrophobic region here and cause partial positive and partial negative. Similarly here, if you have an ion as part of the drug and an induced dipole and a hydrophobic group, then this ion can induce the hydrophobic group and cause what's called ion-induced dipole interactions. And it doesn't have to be the drug that's always positive and the binding site that's always have the hydrophobic region. It could be the opposite. Now let's see what impact do intermolecular bonds have on affinity and on biological activity. Well, first, affinity is how tightly the drug binds to the receptor. This means that the stronger the intermolecular bonds, the higher the affinity. And affinity is typically measured using what's called the dissociation constant, KD, which is the concentration of the drug when 50% of the receptors are occupied. It is proportional to potency, which is the amount of drug required to produce a certain effect, and it is also proportional to efficacy, which is the ability of the drug to produce the maximum biological effect. This is just a brief overview, and you will see more details on the definition of affinity, potency, and efficacy. I just wanted to relate how the intermolecular bonds are related to the affinity. I just want to explain how these two are related. Okay, so we have seen how intermolecular bonds affect the affinity. How do they affect the biological activity? The intermolecular bonds are responsible for the induced fit that occurs to the receptor. So if we have the proper induced fit, then we will end up having the signal transduction. And the signal transduction, of course, leads to the biological effect. Now, if we need to design a drug that should produce the same biological effect, then we would design a molecule that should produce the same induced effect that the natural chemical messenger will produce, and thus leads to the signal transduction and the biological effects desired. To accomplish that, this drug or ligand should have the correct binding chemical groups, and these groups should be positioned in space correctly to interact with the complementary binding sites at the receptor, and they should have the correct shape to fit into the binding site. The ligand should bind reversibly and leave quickly. Due to all these features, it is difficult to design a drug that is very similar to the natural chemical messenger. But if we do, this drug would be very similar in structure to the natural chemical messenger, and we would call it a full agonist. In this example, in this figure, we see the same effect of the agonist compared to the effect of the natural chemical messenger we have seen in figure 5 previously. Okay, so what happens if we design a drug that produces a different induced fit? In this case, the receptor is not going to be able to produce the signal transduction and the biological effect we are looking at. And in this case, this drug will be called an antagonist. This drug would bind to the binding site and prevent the natural chemical messenger from binding to the binding site. It competes with it and will, as a result, the receptor will not be activated and will not see the full effect. Antagonists are of many kinds. Some of them bind reversibly to the binding sites, others bind irreversibly. Some of them can produce no effect at all, and others can produce partial effects. So we're gonna discuss all these kinds later on. Based on that explanation before, I'd assume you would figure out which is easier to design, an agonist or antagonist. And based on this, based on your answer, look at the drugs at your pharmacy and see what is the most common kinds of drugs. Do you see most of them as an inhibitors, blocking agents, and antagonists, or do you see most of them as agonists? Okay, so now we know the importance of these functional groups in forming the intermolecular bonds and how this affects the induced fit and leads to the signal transduction. How can we define what function groups are important? So if you have a drug like that and you have these functional groups, how do you know which function groups are important? Because once we do, the function groups that are important, we can modify them and improve the potency, the selectivity, and be able to produce safer drugs. To evaluate the importance of these functional groups, we follow a systematic fashion to determine how these function groups are important, and this way is referred to as the structure-activity relationship. We have seen this in an example in the previous session where we have a molecule and we would change the function group, either alter it, remove it, or mask it, and then see the effect of that on the activity, and then based on the results we can decide on the important function groups. Once we know what are the important function groups, then if we identify the relative positions in space for these function groups, we define what's called a pharmacophore. So in this particular example, we modified the OH group here with this group, and the activity was reduced, which means it is important here. And another modification was to replace this function group with this, and the activity was unaffected, so we concluded that it is not important. And we repeated the same process for this group, and then we realized that these three moieties or function groups are important for activity, and if we define the relative position for them in space, we can define the pharmacophore groups for this drug. So we have seen how we defined the pharmacophoric groups in the previous example. So here this figure shows the same pharmacophoric groups we defined in the previous example. We have the oxygen, the aromatic, the benzene ring, and the nitrogen, and then if we define the relative positions of these three groups in space, we call this the 3D pharmacophore. There is another kind of a pharmacophore called the generalized bonding type pharmacophore. In this type, we define the interactions that each of this group can be responsible of. So instead of defining an oxygen here, we would say this oxygen is responsible for making a hydrogen bond acceptor with the binding site. This aromatic benzene is responsible for making van der Waals interactions. And finally, this nitrogen, the quaternary nitrogen, is responsible for making an ionic interaction. Once we define the type of interactions, again, we define the relative positions of these interactions in space. This form here is called the generalized bonding type pharmacophore. In this example here, we defined the three most common types of interactions. I mean, we used the three most common types of interactions, but it could be any other interaction that any of the other interactions that we defined earlier in the intermolecular bonds. Notice that in addition to defining the distance between these functional groups, we can also define the angle between these arrows or distances to be certain of the positions. Now, why would we bother defining the bonding type pharmacophore rather than the normal three-dimensional pharmacophore? It is because this kind, the generalized pharmacophore, is more useful in drug design and discovery. The reason is, instead of being strictly tied to the type of the atoms involved, instead of saying we should have oxygen, we should have an aromatic ring here, a benzene, and we should have nitrogen and ammonium group, I could actually say that I need a hydrogen bond acceptor here, which could be an oxygen, a nitrogen, a fluorine, and I need a function group that can make a van der Waals interaction here, which could be a benzene ring or maybe a naphthalene ring. And here I need a positive ionic group. So this is more general definition that could be used to describe the pharmacophore for the active molecule. Another way of defining the pharmacophore for a number of active drugs is using the similarity principle. What does the similarity principle state is that similar biological activity, drugs that have similar biological activities and bind to the same receptor should have a common features among them. These features are defined as the pharmacophore. Let's look at this example here. We have three drugs. They have the same biological activity. They are all of them are painkillers, and they bind to the same receptor. This means that there must be some common features among all these three molecules, and these common features can be used to define the pharmacophore. In this case, you will see that these, the aromatic with the OH here, is occurring in all of them. The same for the nitrogen here, the quaternary or the ammonium nitrogen. You can imagine if you have more than three drugs, say you have 20 drugs, it will be difficult to find the common features, and that's when you will need the use of computer programs to perform that. Especially if you're trying to find the generalized type pharmacophore. When we study the structure-activity relationship for a drug, as we have seen earlier, we identify the physicochemical features that affect pharmacodynamic properties such as efficacy, potency, and affinity, or pharmacokinetic properties such as dissolution, solubility, absorption, distribution, metabolism, and elimination. Therefore, we shouldn't be surprised if a minor change in a chemical structure can cause a dramatic change in the ability of the drug to bind to the receptor, which is pharmacodynamic properties, or maybe the ability of the drug to cross the blood-brain barrier and reach its target receptor, which is pharmacokinetic property. In the rest of today's lecture, we will learn about the physicochemical features that can affect the pharmacodynamic properties, such as steric factors, size, shape, stereochemistry, and conformational change, and other structural features. Later on in future classes, you will learn about the physicochemical features that impact the pharmacokinetic properties, such as particle size, dissolution constant, and the partition coefficient. Okay, so let's look at these structural features and see some examples of how they can impact the pharmacodynamic properties, how they can impact the binding and thus the biological activity. So first, let's give an overview. I'm sure you have seen that in your organic chemistry class, but let's just look at a quick overview at the structural features for a drug. Typically called isomers, can be divided into constitutional isomers, which would have the same molecular formula but will have a different connectivity. For example, the C2H6O has the same one molecular formula that you could derive many structures that could be connected in a different way. These are called constitutional isomers. And then you have the stereoisomers, and there are three kinds of them. There's the optical, which are able to rotate the plane polarized light, such as the enantiomers, the dextro, levo, R&S, and the diastereomers. And you have the geometric isomers, such as the cis and trans. And finally, the conformational isomers, which results from the rotation around a single bond. We will look at the conformational isomers, geometric isomers, and optical isomers. We'll see how these features can impact the pharmacodynamic property of the drug and the ability of the drug to bind to the receptor, make the proper induced fit, and the signal transduction that is responsible for the biological activity as we described earlier. So the first feature we're going to look at is for the optical isomers. We're going to see how optical isomers can affect the biological activity. And let's take enantiomers as an example. An enantiomer would be a molecule that will not be superimposable to its mirror image. So here's a molecule, and here is the mirror image for this molecule. So you could imagine having a mirror in between here, and you can see that one of them is the mirror image of another. And if you try actually to superimpose this molecule into that, you will not be able to do that. So that's why it's defined as an optical isomer, actually defined as a chiral molecule in this case, which is an example of optical isomers. Now, why do you think these two molecules will not be able to have the same biological activity for some receptors? To answer the question, let's assume that this is our receptor. So this receptor will have the binding site for this group here, another binding site for this group here, and another binding site, a third binding site for this group. And you can imagine that all these groups will fit tightly into this binding site and hopefully produce the biological effect. On the other hand, if we take the mirror image for this molecule and we try to fit it to the same receptor, see, this is the original receptor, this is the same receptor, and we try to fit the mirror image of this molecule into this receptor, you will not be able to fit more than two of these groups into here. So maybe you could put this group here and if you rotate this and put it here, the C group will be off, and it's not going to bind. So you will have at least, you can have a maximum of 2 out of 3. So you will have a weak binding, and then you might not be able to induce the same induced fit and reduce and produce the signal transduction and the biological activity. Let's look at a real example here. This is the structure for the ibuprofen, which is a powerful painkiller. This is the left-handed, the S isomer form of the ibuprofen, which is more active than the R isomer, simply because the R isomer is not able to inhibit the what's called COX enzyme, and thus is not able to kill the pain. We're going to talk about COX enzymes and inhibitors later. So this is just an example of how optical isomers can impact the binding to the receptor and thus the pharmacodynamic property. The second structural features we will talk about is the geometric isomerism. In this example here, we have the structure of the estradiol, and we also have the structure of the diethyl stilbestrol drug, which has 2 geometric isomers, the trans isomer and the cis isomer. Now, after knowing that the trans isomer is active while the cis isomer is not active, and we look at the distance between the OH groups here in the trans, and we find that it is equivalent to the distance between for the OH group and the estradiol, while the distance here is not equal to the distance in the estradiol. We realize that the receptor must have some binding sites to fit these OH groups, and the distance between these binding sites should be 12.1 angstrom. That's why the trans isomer is active while the cis is not, because the trans is able to fit the binding sites for the OH groups, while the cis does not. The third structural features that we will talk about is what is called conformational isomers. Conformational isomers are stereoisomers that result from the rotation on a single bond. In this structure here, we have a drug called, a natural chemical messenger called acetylcholine, and we are going to study the effect of the rotation around these single bonds, which will result into having what's called conformations. We will find that this natural messenger, binds into 2 different receptors. One is a nicotinic receptor, another is muscarinic. As we will see in the next slide, studies have shown that the gauche conformation is the form that binds to the nicotinic receptor, because the distance between this quaternary ammonium group, nitrogen, and the acetoxy group would fit the nicotinic receptor. On the other hand, the anti-conformation will produce a different distance between these 2 groups that would fit better the muscarinic receptor. Here is the same structure again, but when we use a Newman projection and we look at the different ways these groups can rotate around a single bond, we will have 4 different structures. We will see that the gauche conformation will produce a distance between the acetoxy group and the quaternary ammonium group for both this and that, that fits the nicotinic receptor, while for the muscarinic receptor, the distance between these two um, is better to fit the muscarinic. So this example we will study in class, and we'll produce the receptors and the molecules to see how different conformation can bind to two different receptors. So different conformations of the same molecule can affect the distances between the function groups and thus affect where the molecule is going to bind. Of course, in this case, the acetylcholine binds to both receptors, but it binds to each one using a different conformation. So if you can imagine, if you want to design a drug that binds to the muscarinic receptor, then you would design one where these two function groups would have the same distance as the anti-conformation. But if you want to design one for the nicotinic receptor, then you would design a drug where these two function groups have the same distance as in the gauche conformation.